presenter will be Sanjay K. Banerjee, National Institute of Pharmaceutical Education and Research, India. Sanjay?
Yes, I'm
Sanjay.
here.
Yeah.
Yes, I'm here.
So, can you just share your screen now?
Yeah, I do that.
And Dr. Sanjay's presentation is on a potential target for diabetic cardiomyopathy. Sanjay, that's fine. You just open up your presentation slides.
Yes. Yeah. Can you see me, my slides?
Yes, that's fine. I'll go ahead with the presentation, Dr. Sanjay. Thank you.
Okay. Thank you, Dilip. Good morning, good afternoon, good evening, all of you. Today, I'm going to present start on a potential target for diabetic cardiomyopathy. So my previous speaker, most of them, they talk about in silico drug design, structural biology, and those are very important for identify how a ligand or drug molecule bind to the particular protein. But before that, we have to identify the target. So here actually I'm going to speak about how to identify a target and how to validate them by using different cell biology and animal models. So as you see my title, I'm going to talk about diabetic cardiomyopathy. So last several years, my laboratory is working on understanding in cardiac complication or or cardiac disorder in diabetes. And if you see, there are some drug available for heart failure patients. There are drug available for diabetes, but there is no drug for treatment for diabetic cardiomyopathy. So before I start my presentation, I'm going to talk about what is cardiomyopathy. Because who are not in this, in cardiac disease field or who are not disease biology, for them maybe the word is new. So cardiomyopathy is a disease of heart muscles. So in generally, it could be due to two reasons. One is, genetic defects in some cardiac protein, mostly contractile protein, and that causing the problem of the heart muscle to contract properly. And or otherwise some kind of factors like hypertension, diabetes, they also altered the cardiac function. So so ultimately the heart muscle change with time, and we didn't define them as cardiomyopathy. There are different kind of cardiomyopathies there, but here my topic of interest is diabetic cardiomyopathy. So yeah, as I talk about diabetes, is a kind of disease which is rise day by day, and in the world, total 422 million adults have diabetes, and there are actually almost 5 million diabetes cases annually. And out of these, 77% diabetic patients are coming from low and middle income countries. But how people die in diabetes. So diabetes, as you know, this is a disease where your blood glucose level is very high due to two reasons. One is insulin resistance, or if you have any problem in the pancreas, which could not able to release enough insulin level. But with time, what happened? You get a different kind of complications. And because of those complications, you get stroke, blindness, heart attack, kidney failure, or in amputation of the leg. So those are the different kind of complications you get with time. But 70% of the diabetic patients, they die in cardiovascular complication. So the major problem of diabetes is cardiovascular complication. So if you look actually what happened to the heart when are you in diabetes, there are several kind of etiological factors and changes are happening in the heart with time, like oxidative stress, altered insulin signaling in heart muscles. There are change in microRNA levels, lipotoxicity, impaired calcium handling, renin-angiotensin systems. There is a fibrosis, advanced glycogen end product change, endoplasmic reticulum that also alter, increased fatty acid utilization, mitochondrial dysfunction, autophagy, apoptosis, inflammation, So there are several kind of changes are happening in heart during diabetes. But another important change which people actually don't thought much is a PTM, post translational modification in protein. So a lot of protein in heart that modified because of some kind of changes, and I'm going to talk like this. So those changes are different kind of, like you can see the palmitoylation is one kind of changes, then ADP ribosilation, succinylation, sulfonylation, and crotonylation, and so many methylation, um, nitrosilation. But in case of when you talk about diabetes, as there's more glucose in the blood, so glycosylation is a major changes happening in the protein, and a lot of people have worked on glycosylation. But one of the important changes happening very early in diabetic state is acetylation in the protein. So why we are expecting more acetylation? Because in early stage of diabetes, you're having a very high level of glucose in the blood as well as free fatty acid level. And because of those glucose and free fatty acid get access to the cardiac muscles, and they utilize those substrate and makes more acetyl coenzyme A through glycolysis pathway. And those acetyl coenzyme A, they easily modify the protein that present in the heart. So acetylation changes in the protein, very early phenomenon that happen in diabetic heart. So as you've seen in this slides, and in diabetic condition when there's an insulin resistance or impaired metabolism, so acetyl coenzyme may basically they modify the protein with the help of acetyl transferase, and then a lot of protein they form acetylated protein. And but in the cells there are some enzymes which always work to deacetylase those protein. Those are called HDAC, histone deacetylase enzymes. So there are different types, class 1, class 2A, class 2B, class 4, and class 3. But more important here, class 3. As you've seen, this is a histone deacetylase. So most of the protein, as I talk, they basically work to deacetylase the histone protein, which is present in DNA. But class 3 protein, which is a called also sirtuins group of protein, which not only do histone, but they do a lot of non-histone protein. That means they are responsible for deacetylase a lot of protein which present in nucleus, cytosol, and mitochondria. So if you look, there are 7 different types of sirtuins are there. So in nucleus, there are 3, SART1, SART6, SART7, cytosol, SART2, and mitochondria, SART3, SART4, SART5. So those are, present in particular compartment of the cells, and they always working to, deacetylate the protein so that the cells can function properly, metabolism can happen properly. So they are play a very important role. So during this deacetylation step, the sirtuin and they activated by NAD, and when NAD bind to sirtuin and they bind to the protein, acylated protein, and then deacylates the protein. So in general, when it is the acylated state, the protein is inactive or sometimes cause a lot of other problem like NF-kappa B is a inflammatory transcription factor. When it acetylate, it binding more in the DNA as a transcription factor, and it causing more increase of IL-6, TNF-alpha, a lot of other, inflammatory gene. So ultimately, acetylated protein in general, not considered as a good, okay? And we have shown previously that, NF-kappa B acetylation happening in diabetic heart, that could be one of the reason for inflammation. So now if you look those here in this TCA cycle, because as I talk about glycolysis and TCA cycle, both are very important for cardiomyocytes because they need a lot of energies and they're very active this metabolic pathway. And several actually enzymes you can see in TCA cycle here, IDH2, GDH, and SDH, SAD2, though those are basically very prone to acetylation. So when they're acetylated, their function become reduced. So we always need sirtuin to be work out here and to deacetylate those protein. In mitochondria, as we talk about, there are total 3, SART3, SART4, SART5 present. But till now, we know that SART3 play a major role to deacetylate all this protein. Similarly, in nucleus, you can see that SART1 play very important roles. A lot of transcription factor as well as co-activator of protein like PC1 alpha, FOXO1 and HIF1 alpha. Those are deacylated by SART1. So those SART1 and SART3 play a very important role to control this cellular metabolism and energy metabolism. So we already knew, and even from our own study, we found that in diabetic heart, there's a decrease, activity as well expression of SART1 and SART3. At the same time, two important, transcription factor, PC1 alpha is a co-activator of several transcription factor, which also play important role of mitochondrial biogenesis, which was downregulated. At the same time, TFAM is a mitochondrial transcription factor present mostly in mitochondria, expressed from mitochondrial DNA, that also downregulated in diabetic heart. So ultimately what happened in diabetic heart, we have seen there is a reduction of cardiac function and left ventricular systolic function and contractility that deteriorated, and there's an impaired left ventricular diastolic function. So ultimately the heart don't pump properly to to supply the blood throughout the body. So we believe that mitochondrial, disorder or mitochondrial dysfunction in diabetic state cause a very important role to cause this diabetic cardiomyopathy. So hypothesis is that now how we can improve the cardiac function. So we hypothesize that activation of SART-wins, especially SART1 and SART3, may improve mitochondrial health and enhance cardiac function in diabetes. So considering the hypothesis or objective to explore the role of SART-win modulation on cardiac mitochondrial health in diabetic rat. So we ask a questions, how important SART1 and SART3 for maintaining mitochondrial health? And for answer the questions, we did two different experiment, and one experiment with the in vitro cell H9C2, which is derived from rat ventricle and is called cardiomyoblast, to explore the role of SART-win modulation on mitochondrial health in this cardiomyoblast. And second kind of experiment we did explore the role of SART-win modulation on mitochondrial health in diabetic rat heart. So the first experiment, as I talked about in vitro test with H9C2 cells. So what we did, we treated this in the cells with the SART1 activators. The molecule is a very well-known SRT1720. Then we also treated the cells with SART1 inhibitors with a molecule EX527. Similarly, we have been treated with SART3 activators, Orexin A. We didn't find any SART3 inhibitors. So what we did, we have knocked down in the cell with shRNA. So we make a stable SART3 knockdown cells, and we measure different kind of parameters or mitochondrial function, looking mitochondrial biogenesis, mitochondrial respiration, mitochondrial dynamics, even oxidative stress. So if you look actually how in general, nucleus and mitochondria, they, link each other, and how they, like communicate by signaling pathway, and through this PC1 alpha, NRF1, TFAM, those gene expression, they makes the protein, and those protein TFAM, and those enter to the mitochondria, and then it enhances the mitochondrial biogenesis. So the first experiment we did to look actually, if we give a SART1 activator, SRT1720, what happened to the expression of SART1, PC1 alpha, TFAM, and SART3. So if you look this data, there's clearly shown that SART1 activator itself increases the protein expression of SART1, and also increase the TFAM mitochondrial transcription factor, which helps to increase mitochondrial all complexes, to increase the mitochondrial respiration. At the same time, SART3, that protein expression also high. But if you treat with the SART1 inhibitors, EX527, at the same time we found there's a decrease of SART1 protein expression, there is a significantly down PC1 alpha, which is considered as one of the important coactivator of mitochondrial biogenesis. TFM, mitochondrial transfer factor, also down. Similarly, SART3 down. So data clearly shows that SART1 can control SART3. When SART1 is activated, SART3 protein expression high. When SART1 is down, and SART3 protein expression is down. So there are links between SART1 and SART3, which present in between two different compartments of the cell, and they control the two important parameters are in the mitochondrial biogenesis and mitochondrial respiration. This PC1 alpha and TFM. So what we did, we also measure mitochondrial respiration by using an instrument. It's called CHOTS. So if you put the cells in the in a plate, and in this instrument they can measure different parameters called basal respiration, ATP production, then maximal respiration, and spare capacity. So if you see the actually graph below, and there are three different kind of graphs is there, where the control group is a DMSO, who is showing the graph. If you treat it with SART1720, SART1 activator, mitochondrial respiration is high. If you inhibit with SART1 inhibitors, EX527, mitochondrial respiration is down, which clearly shows that SART1 activation and inhibition control the mitochondrial respiration, which is clearly tells, yes, there are communication between SART1 and SART3, and that communication works in cells. Now we measure. Next we did experiment to knock down SART3, and then we wanted to see what happened in the cells. So you can see that when you knock down, there's a almost 80% decrease of SART3 mRNA expression, almost 50% the protein expression. And when we measure the PC1 alpha, which is significantly down, but TFM was not changed. Similarly, when we measure the protein expressions, again PC1 alpha is down, TFM didn't find any change. And but interestingly, when we knock down SART3, we found phospho-MPK level is down. So somehow, there are some link between the SART3 expression in the mitochondria and phospho-MPK level. So to confirm that, yes, SART3 control phospho-MPK, we did one experiment, which the data I'm not showing here. If we overexpress SART3 in the cell H9C2, so we have seen the phospho-MPK level is high. So which tells that SART3, they control phospho-MPK level, and phospho-MPK level, they control PC1 alpha. Okay, that's the way the circuit, that on. So from mitochondria to phospho-MPK to SART1, and again it through PC1 alpha and SART3, it activates mitochondria. So if you look at the mitochondria, there are, mitochondria has their own DNA, but only 13 protein of mitochondrial protein comes from mitochondrial DNA. Rest of the protein come from nucleus. So if you look, there are 5 complexes there in mitochondria. Out of these, 7 protein of complex 1 out of 37, 7 comes from mitochondrial encoded DNA. Similarly, for complex 2, there is no mitochondrial encoded protein, but complex 3 only 1, complex 4 there are 3 mitochondrial protein comes, and then complex 5, 2 of the protein comes from mitochondrial DNA. So now what we wanted to see that what we have choose 4 different mitochondrial protein and measured their expression if you knock down SART3 in age-negative cell. So we found that there's a significant decrease of a complex 1, complex 2, complex 4, and complex 5 protein if you knock down SART3. That clearly tells that SART3 control mitochondrial complex protein. And we measure gene expression level of some of the mitochondrial genes which comes from mitochondrial DNA, and all of them look significantly down. Those gene expression when you knock down SART3. So another interesting things that we measure how the mitochondrial dynamics change with SART3 knockdown. So there are, when cells is under stress, there are two important phenomena happening. One is called mitochondrial fission and it's called mitochondrial fusion. So in general, mild condition stress, so two mitochondria they fuse and they make a weak mitochondria. So there are some protein MFN2, MFN1, OPA1, they play a very important role in this process of mitochondrial fusion. Similarly, FIS1 and DRP1, two important proteins which play important role for mitochondrial fusion. So when is a very high level of stress, and then mitochondria itself divide into two, make a smaller mitochondria, they are not much active, they could not make a proper ATP. So in general, mitochondrial fusion is a good phenomenon. It's supposed to be increase more ATP level, but mitochondrial fission is not good. It reduces the ATP level in the cells. So we measured that mitochondrial dynamics protein. We found that DRP level is high, which is mitochondrial fission. So mitochondria looks like divide more during when SART3 is much less in the cells. But mitochondrial fusion protein MFN2, which is downregulated, which clearly says that the mitochondrial dynamics shift towards more fission, which is not making more energetic cells. And we see the similar those changes in protein level, and we measure in certain other parameters, like as you see that SART3 present in mitochondria and superoxide dismutase important for enzymes which neutralize superoxide molecule and play a very important role to reduce ROS generation in mitochondria. So which is more acetylated if you have knocked down SART3. So the protein level is very high, which tells that this acetylated SOD2, we measure their activity, which is much low. So which tells us that, okay, SART3 knockdown, they really, makes the cells to, or mitochondria to generate more reactive oxygen species. And when you measure the whole cells, the acetylated protein, we found that compared to control, SART3 knockdowns have much more acetylated protein, which is measured by the antibody, anti-lysine antibody, and which was expected because SART3 knockdown, they having a less SART so they have increased more acetylated protein. And when we did that mitochondrial respiration in SART3 knockdown, you can see drastically reduction of mitochondrial respiration. So all the data very clearly tell that, okay, that mitochondria is controlled by two important SART1 and SART3. SART1 basically controls SART3, but if SART3 is down, and definitely mitochondria cannot function properly, they cannot produce enough ATP level, or their complex is down, their ROS production will be much high. So now we know that statins are very important for diabetic heart. So how we can enhance the mitochondrial function by a proper treatment? Like now, if you think about treatment strategy, now my data telling that if you activate SART1 and SART3, so chances are more that your cardiac function may be back in diabetic condition. So considering this hypothesis, now we knew previously that there are several pathways is affected during in diabetic condition. Few of them are, as I discussed, mitochondrial function, mitochondrial ROS generation, inflammation, fibrosis. I'll show you some of the data related to these pathways. And so our design was like this, like in animal experiment, there are a total 6 groups. So one is control diet, with the rat given that normal chow diet for 20 weeks. Then we have given a high fructose diet. We choose a high fructose because high fructose diet is not causing more obesity, but it causes insulin resistance. So it's causing more type 2 diabetes. So if we give it 20 weeks, it definitely causing a type 2 diabetes and insulin resistance. Now the next 4 groups, we giving a treatment. So what we did, up until 12 weeks, we giving a high fructose diet. So we have seen after 12 weeks by echocardiography, there is a reduction of cardiac function. But we want to see if we're giving a treatment last 8 weeks with a SART1 activator like SRT1720, SART3 activator orexin A, and also the combination of SART1 and SART3 activator, and with the standard drug metformin. So 4 different treatment we have given, and like to see how this rat behaves and whether cardiac function have any change after this treatment. So in general, blood glucose level was slightly high, but if you look into the interperitoneal glucose tolerance test, which tells you whether they are insulin resistance or not, see there's an increase of the insulin resistance as measured by interperitoneal glucose tolerance in the curve, the area under the curve, which was which was significantly high in this diabetic group, but all the treatment group was down, even better than the normal groups, which tells that there is a reduction of insulin resistance. Similarly, in type 2 diabetes, there's a high level of insulin, serum insulin level, which is we've seen in this red color bar graphs, and significantly down in SART1 activated groups. And also we have seen there's a reduction in other 3 groups. So HOMO-IR, which is a parameter for insulin resistance, again saying that SART1 is a better molecule, SART1 activator compared to SART3 or the combination or the metformin. To confirm that whether same thing happening in cell line, we're giving a H9C2 cells, the cardiomyocyte with a palmitic acid. If you treat palmitic acid for 24 hours, the H9C2 cardiomyocyte cell become insulin resistant. They cannot take a lot of glucose. Their uptake will that reduce. But if you give a treatment of SARTwin as well as metformin, they improve that glucose uptake. Now coming to the histopathology part, and here clearly you can see that high fructose diet, there's an increase of perivascular fibrosis, all the blue color showing that this is a collagen deposition which can see into surrounding the blood vessels. Similarly, interstitial fibrosis, like in between cardiac muscles, a lot of blue color, which are all kind of fibrosis present. But in the treatment group, like SART1 activator, SART3 activator, even the combinations, metformin, all of them, there's an improvement of this, cardiac histopathology. So that we know that cardiac fibrosis, when happen, then there's an increase of matrix protein like MMP2 and MMP9, as well as there's increase of collagen 1. We found that MMP2 is not increased much, but we found there's an increase of MMP9 and collagen 1, which was decreased all of the treatment group. And the inflammation, which is a very common phenomenon during cardiomyopathy, we have seen there is an increase of IL-6 and TNF-alpha that measured by ELISA kit. And then also here serum IL-6 and serum TNF-alpha, which was significantly high. And most of the treatment group, there is a reduction. And when we did the gene expression analysis of IL-1 beta, TNF-alpha, IL-6, similarly, we found there's an increase. Although there are difference in their response of the treatment, but mostly all the SARTwin group and the combination, they work very well, and there's a reduction of inflammation of the SARTwin activator. Now, when you're giving a SART1 and SART3 activator, how the improvement of their own protein. So if you can look that SART1 activator, they increase the SART1 level at the same time SART3 level also. So similarly, when we measure the gene expression and again SART1 activity, SART3 activity, if you look the SART3 activity, so if we give a SART1 activator, so there's an increase of SART3 activity both in a gene expression level as well as the the protein level and also in the activity level, which clearly tells that SART1 can control or enhance the SART3 activity. So few parameters like PC1 alpha, mitochondrial, which is important for mitochondrial biogenesis, NRF1 and TFM, those are important transcription factor for mitochondrial function. Most of them are improved after SART1 activator and also SART3 activator. And similarly, when we measure that transcript level, that also shows improvement after this treatment. Mitochondrial DNA content, which also tells you the mitochondrial number in the bar graph in the C. There's a decrease in the diabetic heart mitochondrial DNA content, which was improved after SART1 activator, SART3, even the the combination group, but we have not seen any improvement, further with the combination. So it's not a kind of any additive or synergistic effect when you're giving a combination of SART1 and SART3 activator. And coming to the mitochondrial dynamics or mitochondrial fission and fusion protein, there are improvement or of MFN2 after SART1 and SART3 activator, as well as um, uh, but we have not seen other protein like DRP1, OPA1, and MFN1. Only mitochondrial fusion was MFN2 was fusion protein was down in diabetic heart, and it was improved with the treatment. So now the last important data, like want to show that how cardiac function improves. So we did echocardiography. So left ventricular diameter, which represent how the big heart, or the inside of the cavity, how big. So one is during diastole, and the second one LVDS. This is systole, like when the heart contract. So during any kind of cardiomyopathy, as the disease progresses, the heart size increases. So there's an increase of the diameter of the heart in the diabetes group. When you're giving a treatment, it comes back to the normal, which tells that treatment basically works very well for the reduction of the cardiac sizes. And another two important data, fractional shortening, which basically tells you how the cardiac the contractility, which was decreased in the diabetes group in the red color and improved in the SART1 activator and then SART3 activator, and also combination and the metformin groups. Similar ejection fraction, another parameter, how much blood throwing to the body, which also decreased slightly around 20% in these diabetic groups, but improve after the all kind of treatment groups. So in general, we have what the data tells me that, okay, we have not seen any big difference, most of the parameter in SART1 activator and SART3 activator. When in try to combine the two activator, we have not seen any extra beneficial effect. Although we have seen improvement, but it was not that additive or any synergistic effect that observed. So now what the summary is that if you give a SART1 activator like SRT1720, it activates SART1, which basically through PC1 alpha increase that gene expression of SART3 and TFAM, which will basically go to the mitochondria and enhance the electron transport chain complex and increase the mitochondrial ATP generation or the activity. And at the same time, through PGC1 alpha, there's an increase of mitochondrial biogenesis. And also we have seen the mitochondrial dynamics, like more in fusion state if you give a treatment. But at the same time, SART1 control the expression of catalase, GPX, SOD2, all antioxidant genes, and in also it SART3 present in the mitochondria deacetylates the SOD2. So ultimately, it enhances the SOD2 superoxide dismutase function and reduce the ROS generation in mitochondria. So overall, mitochondrial health improve, mitochondrial number improve, mitochondrial ATP generation improve, mitochondrial respiration we have seen improve. So those effect directly, indirectly give to the cardiac function. We have seen that improvement by echocardiography. So conclusion is that activation of SART1 and SART3 could be effective to reduce diabetes and diabetes associated cardiac complication. SART1 and SART3 activation together did not show any additive effect when compared to SART1 and SART3 activator alone. SART1 activation can prevent cardiac complication in the diabetic heart by modulating SART3 and improving mitochondrial health via SART1, PGC1 alpha, SART3 TFM axis. And this study actually revealed that SART1 could be a very important and potential target for diabetic cardiomyopathy. Now it should, make some kind of molecule which can activate SART1, and that will be although there are some molecules there, but there are some problem with the molecule. It didn't come to the, although there are some human study also done, but some reasons it didn't come for the human use till now. So this is my groups, and some of them actually generate all the, some of the data that I presented today. And this is my email. If anybody want to contact me in future for any kind of research collaboration, I'll be very happy. Now, thank you very much. I'll be happy to take any questions.
Yes, thank you, Dr. Sanjay. Thanks for the presentation. Any questions from audience?